Mitochondria, often termed the powerhouse of the cell, plays an indispensable role in cellular functioning. The term powerhouse aptly describes the essential function of mitochondria as it generates the primary energy currency of the cell, termed adenosine triphosphate (ADP). This narrative delves into the intricate processes by which mitochondria convert nutrients into energy through cellular respiration, thus sustaining life. The mitochondria reside within the cytoplasm, distinctively bounded by a double membrane, an architectural feature that marks its functional specificity. Within its inner workings, the process of cellular respiration unfolds through a series of complex biochemical reactions. Located in the heart of these processes are the cristae, enfoldings of the inner membrane that remarkably expand the surface area available for chemical reactions. Cellular respiration comprises three principal stages: glycolysis, the citric acid cycle, and oxidative phosphorylation. Glycolysis takes place outside the mitochondria in the cytoplasm, where glucose is partially oxidized and broken down into pyruvate molecules. This stage sets the foundation by preparing the substrates needed for further processing in the mitochondrial matrix through the citric acid cycle, commonly referred to as the Krebs cycle. Here, a sequence of reactions leads to the production of electron carriers, which are pivotal for the final stage of energy production. Oxidative phosphorylation, occurring along the inner mitochondrial membrane, is the ultimate stage in ADP synthesis. Electrons from carriers are transferred through a sequence of proteins, culminating in the reduction of oxygen and the formation of water. The energy from these reactions powers an enzyme, ADP synthase, to phosphorylate ADP, thus generating ADP. This metabolic process underscores the vital role mitochondria play in energy production. By transforming the potential energy from nutrients into usable ADP, mitochondria sustain various physiological activities ranging from muscle contraction to neural activity. In essence, without mitochondria's proficiency in energy conversion, life as intricately woven as we know it would be untenable. In cellular respiration, the process by which cells convert glucose into energy, there are three primary stages: glycolysis, the Krebs cycle, and the electron transport chain. This process takes place within the mitochondria of cells, the primary powerhouse of eukaryotic organisms. 1. Glycolysis * at the outset, glucose, a simple sugar molecule, is broken down into smaller components called pyruvate. This occurs in the cytoplasm of the cell. Glycolysis involves the splitting of a 6-carbon glucose molecule into 2 3-carbon pyruvates, and this breakdown yields a small amount of energy. Specifically, the process results in the production of 2 molecules of ADP, adenosine triphosphate, which act as energy currency for the cell, and 2 molecules of NADH, which will be used later in the electron transport chain. 2. Krebs cycle *. Once glycolysis is complete, the pyruvate molecules are shuttled into the mitochondria, where they undergo a series of reactions known as the Krebs cycle or citric acid cycle. This cycle further breaks down the molecules, releasing additional energy. During the Krebs cycle, each pyruvate is transformed into acetyl-CoA, which is then oxidized to eventually produce ADP, NADH, and FADH2. As a byproduct of these reactions, carbon dioxide, CO2, is released. This cycle operates continuously in the presence of oxygen, contributing to the high yield of energy from cellular respiration. 3. Electron transport chain *. The final and most significant phase of cellular respiration occurs within the inner mitochondrial membrane. Here, the electron transport chain uses the NADH and FADH2 generated in the previous stages to transfer electrons through a chain of proteins. This process is coupled with the movement of protons across the membrane, creating a proton gradient. Oxygen serves as the final electron acceptor, enabling the synthesis of ADP from ADP and inorganic phosphate. This stage is vital as it leads to the production of a substantial amount of ADP, approximately 34 molecules per glucose molecule, by oxidative phosphorylation, ensuring that cellular processes have an ample supply of energy. Through these three interconnected stages, cells can efficiently convert the energy stored in glucose into a usable form, sustaining life processes across diverse organisms.